Welcome to the channel. Let's start with a misconception that costs thousands of lives every year. The most common and deadly form of what we call ovarian cancer does not actually begin in the ovaries. This disease is known as high-grade serous ovarian cancer. Extensive pathological studies reveal that these tumors originate at the fimbriated ends of the fallopian tubes. To understand why this matters, look at the unique plumbing of the female reproductive system. Organs like the lungs or the colon are enclosed tubes. If a tumor grows there, it has to break through a dense anatomical wall to escape. The fallopian tubes, however, are open-ended. They rest right on top of the vast open space of the abdominal cavity. When malignant cells form on these fimbriae, they don't have to fight their way through a basement membrane. They simply detach and shed, like leaves falling into a stream, landing directly onto the peritoneal surfaces and abdominal organs below. Because this shedding process meets no anatomical resistance, the disease seeds the entire abdomen rapidly. The original tumor on the fallopian tube might remain microscopic, while secondary growths spread aggressively across the liver and bowel. This anatomical quirk turns the cancer into a stealth threat. It bypasses the body's structural defenses and spreads in silence, long before a patient experiences anything more specific than everyday bloating or persistent fatigue. The logical defense against a silent disease is to actively look for it. For years, the strategy was to deploy annual screening programs using pelvic ultrasounds and a blood test that measures a tumor marker called CA125. This line graph shows the results from massive randomized trials spanning hundreds of thousands of women, including the UK CTOCS and PLCO studies. It plots the mortality rate of women who received aggressive annual screening against those who received none at all. There is zero divergence between the two lines. Regular screening did not save lives. This failure is rooted in timing. By the time a primary tumor sheds enough protein to trigger a CA125 test or grows dense enough for an ultrasound, microscopic cells have already irrevocably seeded the abdomen. The metastasis occurs long before the disease crosses the detection threshold. Standard imaging and protein markers fail to catch the disease early enough because they are looking for a mass that hasn't formed yet. To move the needle on survival, oncology had to zoom past the anatomy and look into the cell's genetic code. Oncology has shifted toward a molecular framework where epithelial ovarian cancer is defined and treated based on distinct genetic profiles. If we look inside the nucleus of these tumor cells, we find an obligatory early error. The TP53 gene, known as the guardian of the genome, violently snaps and loses its function. Normally, TP53 acts as an emergency brake, stopping a cell from dividing if its DNA is compromised. Without it, the fallopian tube cell replicates continuously, carrying severe DNA damage into every new generation and creating rampant genetic instability. On top of this, up to half of these tumors suffer a second major hit. They lose their primary DNA repair mechanism entirely, a state called homologous recombination deficiency. This is often driven by inherited mutations in the BRCA1 or BRCA2 genes. This broken repair engine is the tumor's greatest evolutionary weapon. It forces the cancer to constantly mutate and adapt at a speed that outpaces the body's immune surveillance. The traditional response to this aggressive growth is maximum force. Surgeons perform extensive debulking to remove visible disease, followed by platinum-based chemotherapy. These drugs are designed to exploit the tumor's instability by flooding it with so much DNA damage that the cells simply collapse. The flaw in this approach lies in the tumor's hypermutability. Under the pressure of chemotherapy, a fraction of the cancer cells will eventually evolve a workaround. They survive, multiply, and return. The cancer's broken DNA repair system creates an inevitable cycle where standard frontline treatments eventually stop working. The fatal threat to the patient is this chemotherapy-resistant recurrence. To break this cycle of recurrence, researchers developed a precision medicine strategy called synthetic lethality. It takes the cancer's broken genetic repair system and turns it into a targeted weapon. This logic matrix illustrates exactly how it works. A normal cell has two active pathways to handle the daily wear and tear on its DNA, a primary repair system and a secondary backup enzyme called PARP. Because the cancer cell has lost its primary repair mechanism, it relies entirely on that backup PARP pathway to survive. When doctors introduce a PARP inhibitor drug, it blocks that specific backup enzyme. The normal cells easily survive this because their primary repair pathway remains intact, but the cancer cells lose their only remaining lifeline, resulting in catastrophic DNA failure. The clinical data confirms the math. In the landmark SOLO-1 trial, administering PARP inhibitors as a maintenance therapy delayed disease recurrence by several years, flattening the mortality curve and extending lives. Because this mechanism is so precise, global clinical guidelines now mandate extensive genomic testing for all newly diagnosed ovarian cancer patients. Doctors have to find the specific genetic signature in order to deploy the exact corresponding weapon. Deciphering these broken DNA repair pathways has allowed medicine to use a tumor's most aggressive trait, its genetic instability, to achieve long-term survival for patients who previously had few options. If you found this breakdown helpful, please like, subscribe, and press the bell icon for more deep dives into the science of medicine.